So welcome, everybody. Good morning, so to say. It's a pleasure to be here. I don't really work in barcoding, as you might suspect, but I do work in phylogenetics, phylogeography, and population genetics, which indeed are very relevant topics for barcoding, especially for those cases which are more difficult. Today, I'm going to focus on a particular topic, which is the estimation of phylogenies, of species trees, when you have many genes. And this might be relevant for barcoding in the near future, right? If you want to analyze data with more than one gene. So what is phylogenomics? Phylogenomics is the phylogenetic analysis of many genes. I mean, molecular phylogenetics started with mitochondrial genes in the early 80s, okay? We were building phylogenies from site V, CO1. But the field is moving, and mainly the NGS explosion of data has changed things. So the field is moving from single genes, a few genes, 10 genes, to thousands of genes. Okay? Nowadays, it's relatively easy to obtain using genome capture, hybrid enrichment approaches, RNA-seq, to obtain thousands of genes for a set of species. Okay? But the problem is still the same. We have sequence alignment. I don't know if you can see the little point. With many genes now, and we want to understand the phylogeny of some organisms. Okay? That's phylogenomics. One of the main discoveries of phylogenomics, perhaps, is the observation that when we use many genes, okay? By the way, I'm using gene in a very loose way. Gene is a piece of DNA, a genomic region, if you want, okay? But I'm gene, locus, genomic region, whatever, it's the same thing, okay? Something that you define what it is, a gene, okay? So when you have many gene trees for multiple regions, probably some of you have already noticed this, you can obtain different answers for different regions, okay? If you analyze different genes, maybe you obtain different gene trees. And this is well known, and it happens at every scale, okay? Happens in a more or less recent scale, 5 million years ago, or less, okay? The history of this species. And you might be surprised or not to know that if we analyze the relationship between human, chimp, and gorilla, only 50% of the genes support a clade human chimp, which we believe is the correct clade, okay? Only 50% of the genes. And this happens all over the place at different time scales for very well problematic nodes of the tree of life, in the Celomata versus Erixothoa hypothesis here. This is the position of, the origin of tetrapods, the Lamphis and coelacanth, and so on. Okay, it happens all over the place, okay? So the main idea is that if you analyze different genomic regions, okay, you might obtain different phylogenetic hypotheses. Okay? So the question is why? So the first and simple answer is to see these are mistakes. Okay? Maybe these gene regions are small. We don't have enough information. We know that phylogenetic methods make both random and systematic errors, right? An example of a systematic error is long branch attraction, as you might know. And maybe what is happening is that some methods are failing for some genes and not for the others. Well, that's part of the explanation, okay? What can we do about it? And I'm going to talk about this in the next slide. Well, we can try to improve our substitution models, okay? Or we try to improve not just the models, but the way we assign models to the genes, okay? I'm going to talk briefly about this next. And we also need to recognize that maybe the problem is very difficult. Maybe some genes are slower than others, and there are, for very short branches in the tree of life, there are no changes for that gene, so it's impossible to recover that node. Or maybe there are a few more places for this gene and not for the other. So there might be errors explaining this, but this is not the whole story. And the main point today of my talk is something that is well known, it's been known for many years, but we are not very aware of this until very recently, is that there are biological processes which explain the disagreement of gene trees for the same species, okay? So first thing, models, okay? So you might know that I work in model selection for single genes. You might heard of model tests or programs like that. But what we do when we have many genes, okay? When we have many genes, you might think of, well, I can run model tests or any model selection tool for every gene independently, and that's it. No, that is not necessarily the best statistical solution. That is not necessarily the best optimal model. When we have many genes, we need to think about them in a joint fashion. We need to assign models thinking of all of them at cell time. So the problem is a combinatorial problem. Is which genes should be assigned the same model of evolution. By model of evolution, I'm talking about Jukes and Cantor, Kimura 2 parameters, okay? HKY plus gamma, GTR. Okay? You know what I'm talking about, right? So basically, a color here is a model. So we have many options. We might assign the same model to every gene, which is there in the top. Okay? We have multiple genes, and we say they all evolve under K2P. Okay? Or the opposite side, the opposite solution would be to assign to each one a different model. Okay? And in the middle, there are many options. So the problem of model selection when we have multiple genes is to find the optimal coloring here, okay? The optimal assignment of models. So basically, how many solutions are there? Many. Okay, these are described by the Stirling numbers of the second kind, in fact, but for 5 genes, there are 52 ways of assigning models, okay? And the number grows rapidly, because for 12, we have 4 million ways of assigning models, okay? And so on, okay? And for 20 genes, which is nowadays is a small data set, okay, phylogenomics, a very small data set, we have, I don't know, what is that number, okay? But a big one, right? So the number of potential solutions to assign models when you have many genes is huge. So other people before us and us, we came out with some algorithms, which I'm not going to explain, but are hierarchical clustering algorithms, similar to what neighbor joining does, which is basically try to find genes that evolve alike and assign them the same color. Genes in the same color, the word we use is they are in the same partition. Okay? So the problem is to find which genes are in the same partition, the partitioning problem. Okay? So we have developed a new tool. You might have heard of Partition Finder, which it's a program that some people use to assign models. It's the equivalent of model test for many genes. Okay? So, of course, we were interested in this problem as well, and we just developed another competitor software, okay, which is Partition Test, which in fact is much faster and it does a little bit better. Okay? So, according to our simulations at least. What can I say? That's the answer for our simulations. So basically here, this is the accuracy scores of finding the right coloring scheme. Because we do simulations, we know the true set of models, okay? So we do a bunch of simulations, 12,000, and we know the true answer, the true colors, the true assignment of colors, okay, of models, and we get this with different algorithms. This is our software, like 30% of the time, okay? 1/3 of the time. You might think, well, this is bad. Well, I think this is absolutely fantastic, okay? Among billions of solutions, okay, 3 out of 10 times we get the right coloring scheme, okay? The true assignment of model. That's, I think, is very good, okay? Well, we are working with only 10 to 50 genes, okay? But still the number of solutions is billions, okay? And these are the settings, and we get the perfect partition recovery 30% of the time. But the good news is that even if we don't get the exact one, we get something that is very close. This is this number. This is called the RAND index, and it measures how well is my model assignment for many genes compared to the true model assignment, okay? And it goes from 0 to 1. So we get 0.97, okay, a little bit better than Partition Finder all of the time, okay? So we are able to identify, in the simulations at least, okay, the true assignment of models for multilocus data sets, okay? But how about the trees, which is probably the important thing? Do we get better trees when we use this strategy, this multilocus model selection? Okay? Well, we do, at least compared to one case, to the case of assigning, okay, one color to all the genes. If you assign the same model to all the genes, okay, in our simulations, again, we get the true tree less than 80% of the time, okay, the perfect true topology. But if we use any of these methods, our program or Partition Finder, we just increase that significantly, right, by 12%. Okay? We do as well as using the true model. This is just using the true model, we get the same accuracy. This is just better than this just because of chance. We are doing simulations. But it might be also surprising that if we do a priori assignment of models, okay, we don't do model selection, we just assign each gene an independent GTR plus gamma, we do also very well, okay? So these are the 2 options you have to analyze multilocus data sets. That's my recommendation. Either you assign a GTR plus gamma model to each gene, or something with gamma will be enough, okay? Maybe for barcoding typical scale, something with gamma, okay? HKY plus gamma to each gene independently, that will be fine. Of course, if you are dealing with 5 or 6 genes, okay, these methods will take seconds, okay, to give you the best fit option. So if you are used to run model tests for single genes, use these programs, Partition Finder or ours, whatever you prefer, okay, to obtain the best fit model for a set of multilocus, okay, data. First, that's the first message, okay? This is a short one. But the most important is this, okay? We know since the 30 years from now that there are two entities when we talk about phylogenies. What is a phylogeny? I said before phylogeny, but I wasn't clear what is a phylogeny. It's a continuum of what? Of genes, lineages. In fact, there are two main entities. And we all the time confound them, okay? And here, you know, in the talks I've been listening, you all the time repeat this thing. You are building gene trees, and you are thinking they are species trees, but they are not. They are gene trees. If you build a tree from CO1, that's a CO1 tree. That's the history of CO1. That is not necessarily the history of the species that you have, okay? Maybe it is, maybe it's not. That's the question, how often it's not and how often it is. Okay. Well, so a species tree is just the history of the species, okay? The history of the speciation events, okay? The wide tree here or there, okay? And we have the history of ABC, and inside, we have the history of DNA replication. That's a gene tree. The history of the DNA replication across parents and offspring through time. Okay? The continuous of the genes, the lineage. Okay. So these are two things. Gene trees live inside the species tree. Okay? But they don't have to be consistent. So if this is a species tree, the dominant paradigm, and in every talk I've listened here, this is what you have been assuming implicitly, okay, or explicitly perhaps, is that the gene tree always follows the species tree, okay? But there are phenomena which explain why this is not. Biological phenomena, okay? One is duplication and loss. Maybe this is not that very important for barcoding because you work with single copy genes. But if there is duplication and loss, okay, so we have suddenly one gene duplicates in red and blue, alpha and beta, they are paralogs, okay? And we have some combination of gene losses, we might obtain a true tree which is different from the species tree. Okay? This is the true gene family tree, and it's different from the true species tree. Other is horizontal gene transfer. And this might be a little bit more important for barcoding, especially for microorganisms, for bacteria. Here, what you have, if there is like a replacement of one gene here because of a horizontal gene transfer event, okay, then the true history of this gene is different from the true history of the species. And finally, the most important, and the one that is being more apparent in the barcoding literature, as far as I had time to take a look, is incomplete linear sorting, indeed, okay? We know that the divergence of the lineages doesn't have to occur necessarily. They have to occur before the speciation events always, okay? Genes have to diverge before the speciation events, indeed. If we are talking about horizontal gene transfer, okay? Between species or migration between populations, same thing. But if we have an ancestral polymorphisms, we have 3 alleles living together, we have these 2 alleles living together, okay, without fixing one or the other through this period of time, then we can have a history, which is true history of this gene, which is different from the species tree, okay? This is incomplete linear sorting. Well, incomplete linear sorting is something well known, and it's responsible for phenomena like, it can be responsible for phenomena like paraphilly, polyphily, or paraphilly, okay? So I've been reading some estimates that paraphilly like this here, look, these 2 guys here from species A are more related to species B. Therefore, the genetic distances are shorter to species B than to the conspecific individuals. So barcoding will fail in this case, right, as far as I know. Is 20% of animal mitochondrial DNA. Those are estimates. Most of the species, the mitochondrial molecule, because it's small, has effective population size, small population size, okay, and rapid evolution, have obtained reciprocal monophily. And this is ideal scenario to do barcoding. Both are monophyletic. Sorry, because you cannot see, this is gone, more or less. Anyway, you have another? Because I need to point places. Perfect. Thank you. Oof, this is heavy. Okay, great. Oh, but I need to do this. Okay. So basically here you have monophily and monophily out there, and that's the ideal situation. But here you have individuals from one species which are closely related to others. Okay, this is a polyphily. And it can be abundant according to some estimates. Another phenomena you might be thinking of, people working in plants, is hybridization. Hybridization is a little bit different from the other phenomena because it creates a loop in the species tree itself. Okay, the species tree, which is the thick thing here, is different, and we don't deal with that in our model. I'm going to explain a model for dealing with this type of things. Sorry, but so far we don't deal with hybridization. Basically, we've been pursuing something like this, which is the full model, okay, to understand the tree of life. Okay? To understand the tree of life, it will be ideal to have a model that considers everything, every phenomena. And we know how to write down this model. Okay? This would be the posterior probability of a species tree, given some parameters that explain the birth-death, okay, of gene families, that explain the effective population size, the internodal times for linear sorting, and the rate of HCT. But computing this is very hard. Okay? Nevertheless, there are species trees methods out there. There are methods out there that try to estimate, species trees directly, not gene trees. One of them that you might be using without knowing is just simply join together all the genes, building a super gene, okay, concatenation, and analyze that as a single gene, a single alignment, and thus you get one tree. Okay? This is called the supermetric approach. And then you assume that by joining genes, the noise cancels out, and then the phylogenetic trend emerges. The dominant phylogenetic trend in the gene trees are due to species history. And that might be fine in most cases, maybe, okay? There are other options, which is just instead of joining all the genes, joining beans, some bean here, another bean, and 3 beans, okay? So you have 10,000 genes, you might end up with 20 beans of genes. So you just have 20 super genes now. This is called binning, statistical binning approach. But for this approach, for concatenation, you are restricted to work with orthologous genes, okay? Otherwise, you cannot concatenate alignments if you have paralogs because you don't know the history of the paralogs, okay? Because we are working in the general problem, okay, we're also interested in methods like the super trees, okay? The super tree idea is instead of joining the genes and building one tree, is getting one tree independently for each gene and then joining the trees, okay? First, you get multiple trees, and then you do a consensus, a reconciliation, a super tree, or you can use some coalescent theory. In fact, these are methods that join the trees thinking of minimum times dictated by coalescent theory, okay, and which are very fast and could be used easily for barcoding, like these ones, okay, which are parametric. And then there are full probabilistic models. Some of you might know the StarBeast, which is the main one, okay, which cares about incomplete linear sorting and nothing else, okay, and assumes this model, okay, which is the multi-species coalescent. The multi-species coalescent is just a standard coalescent that has to live with some constrictions dictated by the species tree, okay. So you have some bounds. This lineage here and this lineage here, okay, they cannot live, they cannot join together. If they cross these pipes, which are the species barriers, okay, that would be horizontal gene transfer. So basically, they have to coalesce here. These guys have to, until here, they cannot coalesce with others, and so on. So you have restriction, okay. So the rate of linear sorting in the multi-species coalescent model depends on the effective population size and time. If you have large populations and short interspecies divergence times, okay, this thing, you have a lot, is very wide, and this time is very short, okay, large populations, rapid speciations, then linear sorting, incomplete linear sorting is very likely, okay, that can happen. Watch out. We know how to calculate these numbers. This is pretty standard. We can calculate the likelihood, the probability of 2 lineages surviving across some time for a given effective population size. That's been known for decades now. And basically, we can calculate this. And this is fine. This is fine for a small number of genes and for just incomplete linear sorting, okay? And it's the way to go, in my opinion, use the full probabilistic models. But computation can be very heavy if you have many species. And there are other processes like HCT, you cannot work with these types, okay? It ignores duplication and loss, which might not be important again for barcoding, but for other questions, for the tree of life, it is important. So we have been working on this. Okay? We've been working, we have developed a method which is now in press in SysBio, it's available online, which is a Bayesian hierarchical model for species trees. And it's semi-probabilistic. So we try to be fast, but do not forget, but without forgetting models. Okay? This is the work of one postdoc in my lab, and it's called Genomu, which is genome in Japanese. So basically, the idea is like this. When we're thinking how to modelize a species tree, we need to consider two factors. One factor is the traditional likelihood calculation, the probability of the data given the tree. This is the first thing likelihood. This is what PhiML, PAUP, PhyLib gives you, okay? The standard likelihood calculation of a tree. You have data, you have a model, and then you can calculate, the likelihood, which is this, okay? But there is other component, which is the important in this talk, which is the probability of this tree existing given this species tree. We can combine this, okay, in a Bayesian model, and then we can calculate the posterior probability of a species tree given data. This is straightforward to extend to multiple locus if we think of them independently. Okay? We just multiply the probabilities. So we have one species tree, different histories for different genes. They might be the same or they might disagree. Okay? And we can calculate the probability of the species tree given the multilocus. Okay? And we do this following some proposal made by Mike Steele and Allen Rodrigo a few years ago, which is called the maximum likelihood supertrees. Because this calculation, okay, this probability in the multi-species coalescence model can take a long time, we can just approximate this with distances. The idea is very simple. We might think, and that makes a lot of sense, that gene trees that are very similar to species trees are very probable, very likely. And gene trees that are very different from a species tree probably never live inside the species tree. And we can model this similarity, the relationship between probability and distance with exponential distribution or whatever distribution we can. Okay? So we have extended this, and basically we consider multiple distances that take into account different biological phenomena. We work with unrooted gene trees, and we work with multiple individuals per species. Okay? So we can do pretty much everything here. Which are the distances we use? Well, these distances reflect biological phenomena. So we use what is called reconciliation distances, the number of duplication and loss you need to invoke to explain this gene tree given this species tree, the number of incomplete linear sorting of deep coalescences you need to invoke given this gene tree and this species tree, and so on. We also evaluate different optimal gene trees root locations during the process and uses other distances that do not model any particular biological phenomena. Okay? I'm not going to explain this. This is the multi-type Robinson-Fold distance, but we try to modelize noise there. Okay? Each distance can have a different weight. Okay? So for some genes, maybe gene duplication and loss distances are more important. For others, it's incomplete linear sorting, and so on. We ignore branch lengths for now. So it's something like this. Okay? This is a hierarchical model, so we have some parameters, some penalties for having many duplications, many incomplete linear sortings, and so on, that genome-wide, and then we can control also independently each gene family. So for this gene family, linear sorting can be very important. For this, duplication is important. For the other, HGT is important. Okay, our model takes that into consideration. The idea, it works like this. I'm not going to explain the details indeed, but basically, it works like this. We have two components, remember, the probability of the gene tree given the data and the probability of the species tree given the gene trees. So we just focus on the second part, which is this, the green part, okay? We delegate the first, in this part, we delegate the first calculation, the calculation of the likelihood of the gene trees, and in our case of the posterior distribution of the gene trees, with other programs. Why reinvent the wheel? If there are programs like Mr. Bayes out there, we just use the output, okay? So the input for our program is the posterior probabilities of gene trees that you get from Mr. Bayes. Imagine you put Mr. Bayes to run with 1,000 genes, you get 1,000 posterior distributions of gene trees, and that's the input for our program. Okay? We do something with that which is called importance sampling. We run a Markov chains Monte Carlo on that to get the posterior distribution of the species tree. And this is the quantity of interest, okay? The posterior distribution of species trees. It's like a Mr. Bayes for species trees. There are other similar programs, but that can give you a posterior distribution of species trees, but they only focus on incomplete linear sorting. We take into account distances that reflect different phenomena. We've done simulations. That's the way we try to benchmark our software. Okay, thousands of them, and we do quite well. Okay, we get better species trees than competing methods. Okay, that they just take into account complete linear sorting. So this is, this is our family with different variations. This is our software. We get very good accuracy for gene trees of different sizes. So I'm not going to go into details. Surprisingly, or not that much surprisingly, we also get better gene trees because the output of our program is not only the posterior distribution of species trees, but also modified distribution of the gene trees. So we get the posterior distribution of gene trees from Mr. Bayes, and in our process, we modify the distribution, and we get better gene trees. It makes sense because we are estimating gene trees with additional information, which is the species tree. Okay? It's a simultaneous estimation of gene trees and species trees. So we get, this is the graph. So this is before running our program, this is after. The frequency of the true tree in the gene tree is much higher. Okay? So we modify the Mr. Bayes output in the light of species trees. Okay. You might think, wow, but this program does so many things, has to be very slow. It's actually very fast. And that's the trick of using distances. Okay? Well, very fast in my opinion. Why? Because in average simulation, and we have 80 species and things like that, okay, 100 genes, 50 genes, it can run in one processor, and the program is parallelized, okay? It can run in a cluster, it can run in 1,000 processors. It takes 1 hour and a half on average. That's not that much. And to give you an idea, okay, this data set with 37 mammal species and almost 500 gene families, it takes less than 6 hours in one processor, okay? All the computers you have here have more than one processor, so it will take less time, okay? You have 4 cores or 4 processors. It will take 1 hour and a half maybe to run this data set, okay? So it's fast, okay? Well, and now, this might be important for you or not, what I'm saying, so I'll just summarize some ideas. I'm not an expert in barcoding, so I might be saying things that are not wrong, but it seems to me, well, that in complete linear sorting, well, no, this is true. In complete linear sorting, it increases the barcode overlap, okay? So when you have these situations like here, what is happening is that the distance, this is intra, this is inter, sorry. The distance among blue individuals, okay, is very high, okay? And overlaps the distance, okay, between blue and yellow individuals, okay? You know what is the barcode gap, okay? So in complete linear sorting makes that. Not only that, okay? This is just due to having ancestral polymorphisms, lineages, and this is just evolution. This is normal, okay? I've seen talks of people saying, oh, I have this lineage. This is evolution. I mean, evolution, we have multiple lineages per species. It's just a question of sampling. If you sample more, in more places, you'll find more lineages for a given species. So the barcode can only, okay, can only decrease when you have more sampling, more genes, and more geographical locations. That's my take on this. But it doesn't mean that it's very important for barcoding. It might be or not, okay? But there might be, as I said before, this guy is closer to this, so the barcode identification will say this individual is yellow instead of blue, okay? The way it works, unless you do use species trees, and then you will recognize this is a blue guy, okay? And, well, if you have many genes, instead of one, chances for ILS are higher. So I think it can be important. Well, there's been some discussions of species trees in the multi-species coalescence recently. I don't know if you know this small debate, okay? In this, I just came reading this on the plane in this paper, Mark Datan and colleagues. Basically, they say that with the multi-species coalescence, you get better species assignments than with barcoding, but there is a reply saying, well, if you do barcoding with more care, you get the same accuracy, but it still recognizes that. So is it useful, you know? Models like the multi-species coalescent, think about species trees for barcoding. Well, maybe. I think that there are ideas. Okay, there are ideas that you could use software like ours coupled with species delimitation software. I don't know if you know this one by Ronald and Yang. You can treat your species as your individual as a single species. This is an approximation, and run our program. And I think it's clear for everybody that good species tree are still important for barcoding in the sense that you need to know, you know, which are the species and the barriers and which individuals are labeled as this species to build a reference library, you know, to know the true species assignment, okay? And you can rely on taxonomic information, or you can rely on genomic information. You rely on genomic information, you should use species trees, not gene trees. So this is the take-home message, okay? Gene trees are not species trees. They can be the same or not. We have model selection schemes for multi-gene data. We have species tree methods that they can have nice features. I'm selling mine. It couldn't be, okay, in a different way. So we have a program that works quite well in simulations that can, by the way, I didn't say this, our program gives you a rooted species tree when the input is unrooted gene trees. And that's very interesting, okay, to find the root of many complicated nodes in the tree of life. So is relevant for barcoding? I think so. Okay, this is the end, the final slide. I think it's relevant. It can be relevant. I think nobody can deny this. It can be relevant. Okay. So species trees, yes. Probably in some cases, you might need to get into estimating species trees for complex barcoding problems. Okay? For species with a lot of lineages, with a lot of ancestral polymorphisms, okay? You might need to go and think about estimating species trees with the data to build your reference library instead of using gene trees. When you have many genes, we're talking about the multiple genes case, okay? Only for one gene might be also appropriate. You can build, by the way, you can build a species trees with one gene, okay? And that doesn't mean you get exactly the same species tree. There are examples out there with one gene, which you use the multi-species coalescent, and the species tree is different from the gene tree. Okay? It's not normal, but it can happen. So I think it is. But the question is, if it's relevant, but how often it's relevant? How much relevant it is? That I don't know, because I'm not familiar with the field. I don't know. I think I can say it can be relevant for difficult cases. I don't know how much relevant. But the more data, I'm going to repeat this, the more data you get, the more genes you get. When you start working with 2, 3, 5, 10, 20, if you need to do so, which I'm not sure, to work with many genes, then you will find this type of problems more often. And the more, and also geographical sample. The more you sample across the geography, you'll find more examples of linear sorting. Okay. So I think this is all. So I will acknowledge my students, which are going now to different places, funding, okay, from Europe and Spain. And thank you again for the hosts for inviting me here and having the opportunity of meeting the Barcode community. Okay, thank you.